Good morning, everybody. Today we're going to talk about Chapter 12, Section 2, which discusses the replication of DNA. So we're going to look at how DNA is able to make copies of itself to be passed on to new cells that need the DNA. The statement here says that DNA replicates by making a strand that is complementary to each original strand. So I want you guys to take a second and think about the things we've already talked about when cell division happens and things start copying themselves to move to the new cell. So think about that for a minute. Think about what you already know. Push pause for about a minute and think about it, and we'll carry on here in a second. All right, hopefully you had a chance to pause that and think about the things you already know. But let's go ahead and move on for today. Here are our main goals or our essential questions from this lesson. It says, what is the role of enzymes in the replication of DNA? How are leading and lagging strands synthesized differently? And how does DNA replication compare in eukaryotes and prokaryotes? So if you can answer these questions when we are finished, you know exactly what you need to know. Okay, first we're going to look at something that's called semiconservative replication. So during semiconservative replication, the parental strands of DNA, which means basically the original strand, it separates, and each strand serves as a template and produces DNA molecules that have one strand of parental DNA and one strand of new DNA. So if you kind of look at this picture here, here's the original strand. It's kind of a double helix, right? So there's two strands cut together to make the parent DNA. Well, it's kind of ripped open, replicates, so the new strands are only half new. It's half new, half new, half old, half old. And then that's going to do the same thing. It's going to rip open, duplicate again. So that's where the word semi-conservative, because some of it's conserved, not all of it. Half of it's new, half of it's old. Now there are three main steps in this process: the unwinding step, the pairing step, and the joining step. And we're going to look at that today. So unwinding, this is the first step in semi-conservative replication. So when unwinding is happening, a thing called DNA helicase, which is an enzyme, it attaches to the DNA and it unwinds the helix, kind of like a zipper. It unzips it, and it breaks all the hydrogen bonds between the bases so that they can come apart. And then something called single-stranded binding protein, it keeps the DNA strands separate during replication. And then finally, something called RNA primase adds a short segment of what's called RNA primer to each DNA strand. While it is unwound, there's an enzyme called DNA polymerase that adds the appropriate nucleotides to the new DNA strand starting from the 3' end. So what this does, DNA polymerase attaches to the unzipped strand, and it goes down the line basically attaching those complementary nucleotides. If you remember from our last lesson, we talked about how adenine always goes with thymine and guanine always goes with cytosine. So if whatever it has, it's going to attach the opposite one that it pairs with, creating essentially a brand new strand of DNA. So the leading strand is built continuously, and the lagging strand is built discontinuously in small little fragments. So sometimes it's a perfectly long strand that's just continually being made. Sometimes it's not perfect, and it makes little bitty strands at a time that are later connected together. Well, these little bitty strands, we have this word here, it's called Okazaki fragments. So when you see that word, it's talking about those discontinuous strands of DNA that are created from the DNA polymerase. Okay, the next step we're going to look at is the joining step. DNA polymerase removes the RNA primer and fills in the place with DNA nucleotides. And then we have a thing called DNA ligase, which links the two sections together. Remember the last slide we talked about sometimes it's discontinuous. So this DNA ligase gets in there and hooks them together to make the long strand of DNA that's required. Now, if we were to compare how DNA replication occurs in a eukaryotic cell versus a prokaryotic cell, we're going to see that it's a little bit different. In a eukaryotic cell, which again, that's going to be our cells, animals, humans, plants, it's going to unwind in multiple areas because our DNA is obviously it's microscopic, but it's really big for what it is. It's really long. Um, so instead of one enzyme attaching and going all the way down the line, many enzymes are going to attach and do small segments that get reattached later. Now, with prokaryotes, it's a circular strand of DNA and it just opens at one end and goes around till it gets to the other end. It's much smaller, much simpler. It doesn't require doing it in segments because it's so small. But that's the difference between DNA replication in eukaryotes and prokaryotes. And that's it. Our lesson was pretty short today. Um, if you need to go back and go over a few things, feel free to rewind this video or to look at the attached PowerPoint. But go ahead and make your way over to the discussion board question for this week. Answer or respond to the prompt, and then before the end of the week, make sure to reply to 2 of your classmates' posts.